Thank you, Dr. Schiefer. Okay, so it's a pleasure to introduce probably the most distinguished person I've ever had the opportunity to introduce. Right? I don't know how many people in the room actually save lives, but that's what he does on I think probably at least weekly basis, on a daily basis. So Dr. Francis Charles Brunicardi, he got his BA at Johns Hopkins, right? So you're from New Jersey originally, then went back to New Jersey for medical school at Rutgers, right? And then spent some time at the SUNY Health Science Center in Brooklyn, where you're resident in general surgery, then a research fellow, senior resident, and eventually chief resident in general surgery. So Dr. Brunicardi has published over 291 papers. He joined us at University of Toledo in 2017, right? And he's currently the chair of the Department of Surgery, the director of our cancer program. I did a quick Google search, and I found one of your videos on YouTube has got over 65,000 or 6,500 views of suturing, right? But it's the one that's in Spanish. It's weird. I don't know. Maybe you were down in Texas or something, right? When you were doing that. It's good for anyone who is an MD. If you would like to know how to suture, go and watch his videos because they're actually pretty good. And you also play rhythm guitar. Yeah, I also saw a session on there too. So pretty Renaissance man, I guess is what we might call him. He's going to talk about the precision cancer therapy platforms and the repurposing polypharmacology type stuff that he's been doing here, and really pushing translational medicine at UT into different strats here, right? So go ahead.
Wow. Thank you, Isaac. That was a nice introduction. I'm honored to be here today. I want you to know that my heroes are the basic scientists. So I'm a surgeon scientist, which is an oxymoron. I just really admire the basic scientists, and what I'll show you today really comes out of a collaboration with basic scientists. And I'm really excited about what's taking place today because we're assembling a precision therapy program at the University of Toledo that we hope will leapfrog over all the other schools. And so what we've heard today and heard last week, Dr. Fedorov, we can put a bioinformatics program together that will greatly speed our discovery of drugs and get them to the patients in a clinically relevant time frame. So what I'm going to take you through is a little bit of history for the students. And so we've been on Earth 7 million years walking upright. And so what we're talking about is this fraction of a second in terms of human existence. So DNA was discovered in the 1860s by Friedrich Miescher, who pulled this stringy substance out of pus. At the same time, a monk in Austria was doing peat plants. He was from a gardening family, and he figured out Mendelian genetics. He wrote an obscure paper. About 50 years later, they did the same thing in a science lab, and they gave him credit. They found his paper. And then you flash forward to the sequencing or the structure of DNA. And so on your left is Watson, who was from Chicago, got his PhD at Indiana, and he wanted to go to Cambridge to work with Crick. And if you read his book on the discovery of DNA, it reads like a dime store novel. It's almost like reading his diary. And it seems to me that they really didn't know what they were doing. They were not experts in any area, but they somehow put it together and figured it out. And this is Wilkins and Rosalind Franklin. And they discovered DNA in 1953. The three on the left, on your left, won the Nobel Prize. And the reason that Rosalind Franklin didn't win it is because she died of ovarian cancer. So was that from the X-ray crystallography, or was it from that she was Ashkenazi Jewish and had a BRCA1 mutation? And perhaps if we had better treatments, she would have won the Nobel Prize. So then you flash forward and you go to the Human Genome Project, which I think is one of the greatest discoveries in human history. And it took 10 centers, 10 years, and it cost $3 billion, right? And so then what an incredible accomplishment. Then you flash forward 7 years, and it took one center 2 months to do James Watson's DNA. He volunteered to do it. And it cost $1 million in 2 months. So that's a 10,000-fold decrease in speed and an increase in speed and a 10,000-fold decrease in cost. So we're talking about exponential changes. Now, Francis Collins, who headed up the project, originally from Michigan, said that it is fair to say that the Human Genome Project has not yet directly affected the health care of most individuals. So, and we still are at that, and I think this is the slide that you could keep in your mind, is that precision medicine, there is a huge gap between all of our omics work that we're doing and clinical care. And what we want to do is to build bridges across that gap. And so we've recruited John Nemanitis here and he's really interested in precision cancer therapy for cancer patients. And he's building this spectacular clinical trials program. And his also expertise is how to commercialize the drugs that you're going after. So we're really, really excited about what's happening. And we want to partner with the basic scientists to move this thing along. So we're going to give you an example. We'll give you an example now. So pancreatic cancer is a death sentence. Nobody has been able to improve the survival of pancreatic cancer in the last 40 years. It's really almost a death sentence. The only thing that works is the red line is if a patient can have surgery, the survival is about 25% over 5 years. Still dismal compared to other types of surgery. But the black line are patients who can't have surgery, which is about 80% of all patients, and they die within a matter of months. And nobody has been able to figure this out, how to improve it. Now, so back in the I guess at the turn of the century, I started working with Richard Gibbs, who runs one of the big genome centers. He was the one that did James Watson's DNA at Baylor. And he had a genome center with about 400 people working in it. And he had gotten like $600 million of NIH fund. He came to me and said, could you collaborate with us? And at first I was like kind of stumped on that one. And what he wanted was he wanted us to bring the human specimens from the OR to the Genome Center. So we were like the pack mules, and we were bringing in pancreatic cancers, liver cancers, and colorectal cancers, and they started sequencing. And it was really an amazing project. So this is what we know now. We're scratching the surface of some of the common mutations. And so the most important, so as the cancer develops from normal through the pannins, it's called, and then to invasive cancer, which is really the death sentence. KRAS is a big one, P53. So those are some of the things that we're working on in the lab. Now, we sequenced 142 pancreatic cancers, and we were all excited. It cost millions of dollars, and we were all involved in it. I learned a great deal from working with Richard and his team, and we got a Nature publication in 2012. Yay! Now here's the problem. People are calling me going, I'm dying of pancreatic cancer, and we know you're doing all this work. So as wonderful an accomplishment as this is, it has nothing to do with taking care of patients. And after a while, you just get tired of people calling you from all over the world, and I say, I can't help you. So we really wanted to go after something therapeutic. And we've been working on this for about 20 years now, so I'll tell you where we're at now. So what we decided to do was to use RNA sequencing to go after overexpressed genes as our platform. So we did a genomic profile with a microarray database and RNA sequencing of normal pancreas versus cancer in the same specimen. And we came up with a network of genes, and Zhuhua Yu did this. I hope Zhuhua will join us at the University of Toledo. And so, as you can see, BRC5 is at the center of this network. And Dr. Liu, who's been my partner for 16 years, felt that BRC5 was a very important target for us to go after. So that's why we chose that target, even though we are looking at the network. Now, last year, John Neumonitis said, Chuck, if you're going to go to commercialization, you have to pick one target. So we did pick BRC5, and you can see it's overexpressed in human cancer. It's also overexpressed in the pre-cancers, PAN3. This is a curable cancer, and then it's overexpressed in the KRAS mouse model. Right? This was interesting. We then did this technique for all 9 major cancers, and then we did it for all 9 major cancers, and we came up with this network for all 9 major cancers. So those are the major players across all 9 major cancers, and we were really excited that BRC5 was a major hub. So we then started looking at BRC5 expression, and it's expressed in almost all cancers, including liquid cancers as well. Now, this is beautiful. Dr. Liu started getting normal ductal tissue from mice, and now we get it from humans out of the University of Miami from a colleague of mine. He then dices it up, puts niche factors in, and creates these stem cells. And up here, he CRISPRed in KRAS G12D and knocked out P53. And you can see how he transformed these normal duct tissues into pancreatic cancer. And so here's the wild type. And we're now using IncuSite in the basement. And you actually can watch this in real time every 2 hours. You watch them transform. And so here's the transform. This looks just like pancreatic cancer. And here's your beautiful wild type organoid. So then Dr. Liu stained it, and here's the staining for KRAS G12D. You see that P53 is knocked out, and there you go, you get BRC5 overexpression. Whereas in the wild type, there's no KRAS G12D, normal P53 is expressed and no BRC5. So within a matter of days, we were really excited that BRC5 was being overexpressed, and it's a reflection of these driver mutations. So what we decided to do, and we've been doing this for a very long time, but we decided to take a different tack. We created a BRC5 super promoter. So this is something that Dr. Liu and I don't know, we figured it out a long time ago with the insulin promoter, and Dr. Liu created one for BRC5. And the way he does this, he takes out he takes a region and he pulls out all these transcription factor binding sites and then puts them together into one gene. We send off, I don't know, 16 clones, and then they're made by a company. They send them back to us, and we then test them. So here is the endogenous promoter of BRC5 driving Gaussian luciferase. Here is your CMV, your ubiquitous promoter. By the way, that was made at the University of Iowa. And then here is our BRC5 super promoter. So we're really able to drive gene expression compared to the endogenous promoter. Then what Dr. Liu did is he supercharged it. He put on 2-step transcriptional activation. And you can see that compared to the regular super promoter, like this would be the super promoter, now he's got exponential gene delivery. So we're using these super promoters for drug screening, as well as we're doing gene therapy for imaging and therapy as well. But I'm going to talk now about our drug screening. So the way we do this is we drop the gene into a pancreatic cancer cell. The transcription factors activate our super promoter that drives luciferase, and then you add a prodrug and you get luciferase. So then what we did, and this was an accident, we wanted to look for novel drugs. So we gave our assay to the core at UCLA, and they ran this core. Well, what they ran was FDA-approved drugs. And I said to the core director, why did you do that? And he goes, I don't know, I thought it'd be a good thing to do. So I started thinking about it, FDA-approved drugs, and I started reading about repurposing. I'm like, holy cow. So what we found is that all these drugs are inhibiting our super promoter in cancer cells. And so my wife and I talked a lot about this with Dr. Liu, and we picked out metformin, digoxin, and simvastatin. Now, what we did, over a year, we did drug screening, and Zhu did a lot of this work with Dr. Liu. So we did single agents, double agents, and triple agents. Now, as a surgeon, triple therapy works. Triple antibiotics, we use it all the time. It's what turned AIDS into a chronic disease. So we picked these 5 drugs, but what we did is we wound up focusing on these 3 drugs, metformin, simvastatin, and digoxin, which had a really, I thought, a synergistic effect on inhibiting pancreatic cancer cells. So why did we pick it? They were safe and non-toxic. It's not toxic chemotherapy. They're off patent, generic, and inexpensive. They cost pennies a day. Some of the new cancer therapies are like $800,000 for a treatment. Their patients are already taking the drugs with no toxicity or minimal toxicity, so we know the toxicity. And each drug separates the pathways. We looked everything up about the 3 drugs are all involved in cancer. And I will show you, all 3 drugs suppress BRC5, which is our cancer target, our candidate. So then we started testing, these are commercial cell lines, and these are the patient-derived cell lines. So we have 16 cell lines directly out of the patient's body. And so you can see the control is here. Simvastatin alone gave us this much suppression of cell viability. Digoxin, interestingly, in the commercial cell lines had a powerful effect, but in the patient-derived cell lines had very little effect. Metformin had very little effect, but you could see they were synergistic in all 4 cell lines when you put them together at doses that kind of resemble what's being used that humans take. So what's interesting about this is you can see that it's almost like precision medicine, that you might have to test this on the patient's cell lines. Dr. Liu's a wizard at the mouse models. So he engineered these cells to give off Gaussian luciferase. So now we have a companion diagnostic in the mouse. You can measure the bloodstream, and you can see the tumors as they're growing, giving off Gaussian luciferase. And you can see the effect of our drugs suppressing our, what would you call it, our synthetic biomarker. And here, this is only over a month. You can see the phosphate buffer control, and the drug was, I guess you would call it cytostatic. I don't think we treated them enough to see the death effect. And then, of course, we can use bioluminescence, and here's our control versus our treated. So we quickly took, and this is, by the way, a patient-derived cell line, and we can see that our 3 drugs actually worked in the mice. We then took those tumors out, and we ran RNA sequencing and put it through weighted gene co-expression network analysis, and we found these cell cycle genes, and we were excited that BRC5 was one of them. And here are the different categories. We did this in 3 different things. We then ran mRNA level for BRC5 to make sure that it was, and sure enough, C3 had a major effect on inhibiting BRC5 mRNA. And here is the C3 effect on the BRC5 in a Western blot, so knocking down the protein. And now these slides I just love. I'll start with this one. You can see that BERK5 just complete C3 knocked down BERK5 in the human tumors growing in mice. And so we're really excited about that. But this is where the C3 is having its effect. It's having its effect at the promoter. And so Justin Creedon is the MD PhD work in the lab. We're so excited about his work. He's going to try to figure out the mechanisms of how C3 is acting, probably on the transcription factor or epigenetics of the promoter. But what this is a like a video showing you over time how C3 is inhibiting the BERK5 promoter, which is driving GFP. So we now have a targeted therapy for pancreatic cancer using BERK5 super promoter to identify FDA-approved drugs, and we picked that combination. The great news, okay, so I'll get to that in a second. So then we had a student, Steve Markowiak. He probed the database at ProMedica, 2.5 million patients, and he pulled out 72 patients who were taking a statin, metformin, and digoxin, and he showed in a control group there was no evidence of any toxicity above the normal toxicity of the patient. What that allowed us to do is John Neumanitis wrote a protocol for a phase 1b clinical trial, and he sent it to the FDA. They gave us an exemption. We then took it to the IRB. We now have IRB approval to take these 3 drugs to patients who have end-stage cancer. So we're really excited. So we think what you're looking at was about 5 years of work. We think we can condense this into a few months now that we know all the steps involved. So then I asked Bill Maltese when he was here, and I really miss him. I said, what are we doing with the super promoter? I don't understand. Like, I know we're doing something, but I don't get it. So he drew this out on a piece of paper. I asked Dr. Liu to do this, and here's the data from it. When you're using an endogenous promoter, which is how most drug screening is done, you might see an effect, but there's too much noise, and so you hardly see the effect. The synthetic promoter, you're saying, okay, you're starting to see effect, but with our super promoter, when you see an effect that's powerful, this is what Bill Maltese said, when you see an effect that's powerful, you know that C3 is really doing something. So I guess you would say this is really highly sensitive. And I think that's how we figured this out. And so this now becomes an assay of testing how powerful your drug is on your target. So in conclusion, we demonstrated a feasible, cost-effective platform for repurposing FDA-approved drugs. Using a super promoter in a clinically relevant time frame. And my last slide is none of this would be possible without a large collaborative effort. And we're really excited that Justin's in the lab getting his MD PhD. Dr. Liu's here. I would love to recruit Zhuhua Yu. He called from China yesterday. And of course, John is here with his team. And so this was the people that generated all this data. Rob, thank you for inviting me. I'm happy to answer your questions.
That's a great talk. 2 questions?
Sure.
Yeah, so we got time for a couple questions. Great talk. I'll ask one if no one has. So there's probably some standard of care the patients are already going to be taking, right? You're saying these are end-stage cancer patients, so they're probably already be given some chemotherapy. Is that right?
They've already failed chemotherapy, so they failed the 2 phases.
So you're not going to have to give anything else in addition to your 3 agents. That's it. Okay.
It's a very simple, clean trial. And we have some evidence. We found a paper that patients who were on statin after pancreatic surgery, they lived an extra 15 months. And patients who were on metformin lived an extra 30 months. So we have some evidence that using the 3 might work. So we're excited to see what happens.
So that was awesome. So how many patients will you enroll in a phase 1b trial?
Okay,
What's the goal there?
I thought we could go straight for a phase 2. So for those who don't know, phase 1 is just to look at toxicity. Phase 2 is to look to see if the drugs have an effect. And then phase 3 is like 300 patients in each arm. John wanted to go with phase 1b to get the exemption from the FDA. He said if you go for phase 2, they're not going to allow it. So we're going to give 3 patients low dose of the 3 drugs, 3 patients medium dose of the drugs, 3 patients high dose. Then we're going to add on an extra 6 patients with high dose, and those patients will have a biopsy of their metastasis, and we will see if we inhibit BRC5 targeting. And so that's the trial.
What would a positive outcome look like?
Well, lack of toxicity is the purpose, but if we prolong life, and all these patients are going to die within months, so if we start seeing them live, all we have to do is have them live 8 weeks, and it's a big success as an average. But that would be phase 1. Then we could roll straight into a phase 2, and let's say do 30 patients per arm.
So we have a couple more questions. Bruce?
Could BRC5 be the basis of early diagnosis for pancreatic cancer?
That's a good question. So that the CRISPR slide where BRC5 went up with the mutations, so Dr. Liu is working on an early detection platform. And the question is, how do you detect it? We have a viral gene therapy for detection, but the reviewers on the NIH don't like viral gene therapy for imaging and detection. So they've kind of blasted us on the grant. So we may try a different delivery system, maybe like exosomes or liposomes. So yeah, we're working on an early detection platform.
Yes, I have one more question. This may be totally off base. I was just quickly checking what iLinks tells us about the fighters, tons of stuff, and then Jim has some presentation later. But metformin, it seems, might inhibit not only BRC5 but also BRC6. So I was wondering if there's any evidence of compensatory expression of some problems.
It's a great question. I'd have to look that up. We're constantly, every day, looking up new things and new ideas, but we want to work with your group to see if we can somehow do this with what was all presented today. And then maybe we could streamline this and just test it on the patient's cells and go fast. Yeah.
So thank you very much. That was awesome. That concludes the sort of platform speaking based part of this.